Welcome again to the lecture series on research methods. Lecture 6 will cover quantitative research and mixed methods. We'll be looking at study designs, sampling techniques, data collection methods, and mixed methods research. You were already introduced to study designs and quantitative research in the first lecture. By way of recap, when we talk about quantitative research, it's the process of collecting and analyzing numerical data using a variety of statistical models to support reporting trends, relationships, and associations. When we talk about study times, they could be described based on the design. And in lecture 5, we focused on qualitative design. And today we are focusing on quantitative research. Research can also be described based on the intervention. And you were already shown the different study types that you can have in lecture 1. So it could be observational and it could also be experimental, in which case you do have an intervention. So we would look at more detail at some of the study designs. The study can also be described based on time. You would hear things like prospective studies, and you would also have things like retrospective studies. This is also recap because you were introduced to this already to show the evidence that can be generated from different studies. Randomized controlled trials are usually said to be the gold standard. From randomized controlled trials, from systematic meta-analysis. These are high quality studies. They are consistent. The results can be generalizable. You would have sufficient sample size for your study, and so you're able to make definite conclusions. If you compare these to other studies lower in the pyramid, these studies they tend to be inconsistent. You would have only few cases in such studies, so you can't really make definitive conclusions from this. So as you go up the pyramid, you see that the quality of the studies improve. We are going to talk specifically about the study designs. So if you remember, we said that cohort study is a kind of analytical observational study. So in cohort study, you are first trying to identify an exposure. So you identify a study population where people are without the disease, and you have two groups, those who are exposed and those who are unexposed. You follow them up in time to see those who would develop your outcome or disease of interest, both in the group that is exposed and in the group that is not exposed, and you compare. If you look at it in terms of timelines, you can have prospective cohort studies where you start with your study population of exposed and unexposed, and you follow them into the future to see who develops disease and who does not. Cohort studies can also be retrospective. So you start at a particular point in time where you have your cohort of exposed and unexposed, and then you also look and see those who have disease and those who do not. But the data is already available, but you're now assessing it in the past to see how people progressed to have disease or not. So there are several advantages of doing a cohort study. Usually you can assess causality because you're gathering data regarding sequence of events. So you're looking forward in time, so you are able to assess causality. You can determine incidence rates and risk factors because you're looking at how things progress. So when new cases come up, you're able to detect those. It can be efficient for studying rare exposures. Cohort studies can also help to examine multiple outcomes for a given exposure. Say, for example, smoking, and we'll be looking at examples shortly. If you look at smoking, you can look at outcomes such as cancers or links to respiratory illness or links to cardiovascular illnesses as specific outcomes. From the cohort studies, you can also eligibility and outcome of the assessments can also be standardized. Disadvantages would include the fact that it can be very expensive. For rare exposures, you would need a very large sample size. The results are not available for a long time because if you can imagine, especially for prospective studies, you're looking at the outcome as they occur in time. So it might take a very long time. There could be tendency for bias to attrition and loss to follow up. And this could affect your cohort because if it's different between the exposed and the unexposed group, that would affect your results. And considering the fact that it takes such a long time, people may drop off and not want to continue the study to the end. An example of a cohort study is a well-known study that occurred in the UK among British doctors. It was a prospective study for a long period, 1951 to 2001. More than 30,000 individuals were followed up. They were examined for smoking habits, disease rates, and mortality. So the exposure here was smoking, and the disease they were looking at was lung cancer or mortality. From the conclusions, they had several follow-ups. They were publishing the results regularly, and they found out that individuals who smoked tobacco were likely to develop chronic respiratory illnesses, including lung cancer. And if you look at the chart, the non-smokers definitely had lower death rate compared to the people who smoked. And the more you smoke, the higher your chance also of dying of risk of the higher your risk of dying from lung cancer. Let's also look at case control studies. With case control studies, you're starting with the outcome. So cohort studies, we start with exposure. Here you're starting with an outcome, so a disease in a particular case. And you now look for controls who are individuals who do not have the disease, and usually they should be from the same population. You would now go back in time to see what risk factors were associated possibly with the disease. So you compare for your cases, those who have the risk factors and those who do not. You compare these to the controls, those who have the risk factors and those who do not. In terms of the timeline, so you are starting with the cases and then you're looking back to see what risk factors people would have had that led to their disease. The case controls could have advantages in that it's inexpensive and can be very efficient, especially for rare diseases. Because if it's rare disease, you don't have to wait for a long time to get your cases. It can be useful for studying diseases with a long lap between exposure and outcome. So it's faster for you because if you're waiting for a cohort study for someone to develop a disease, it may take a long time. But in case control studies, that lap is reduced. You can also use it for multiple exposures or risk factors. For example, if you say a child with asthma, you can have multiple exposure factors that can be looked at for these conditions. It requires comparatively fewer subjects. One difficulty, though, for case control studies is the selection of appropriate control. For example, if you look at diarrhea in children, if your cases are children who present to hospital, where would you collect your controls from? If you take children who do not have diarrhea who come to hospital, they may not be representative of the rest of the community of people who do not have diarrhea, because it may be that it's the very sick children who are coming to hospital, for example. You may also have potential bias in measuring the exposure. because you're looking at it in the past, so people may not remember. There's recall bias. And also, even if you look at it also, the way somebody who's, for example, a mother whose child has asthma may remember more than somebody whose child does not have asthma, may remember more of the risk factors. So these can affect your cohorts, your case control study. We can look at a specific example of a case control study that was conducted here in the Gambia, where the Gambia liver cancer study, they've really conducted a lot of studies. They also have cohort studies. So if you're interested, you can look at the literature. This particular study was looking at risk for hepatocellular carcinoma, and they found that hepatitis B and C were risk factors. So the researchers recruited cases of people with hepatocellular carcinoma, and then they also recruited controls. So the controls were people who do not have cancer. If you look at this, you will see that the number of controls is twice as much as the number of cases. And I'm sure you will go into details when you look at the statistics in your next lecture series. So for this particular series, they found that carriage of hepatitis B surface antigen was higher among the patients who were among the cancer patients compared to the control. I'll focus now on cross-sectional studies. In cross-sectional studies, we're measuring the exposure and the health outcome of the study participants at one time point. So the advantage is that it is cheap and it is really simple study to do. The disadvantage is that though you may be able to establish association, you are not able to establish causality, because you're measuring both the exposure and the outcome at the same time. So you can't really tell which one occurred first. You may also have issues with recall bias, because again, you're asking specific questions on exposure and people may not always remember. The example here is a study that was done here in the Gambia, looking at prevalence of malaria among study participants. So a number of study participants were enrolled, and then you test them for malaria, and they found a prevalence of 16%. They also looked at a sub-study. So among the people with malaria, you look at the percentage that were asymptomatic. So these really are prevalence studies that were conducted here. Like we mentioned initially, randomized controlled trials really are the gold standard for study designs. And so with the randomized controlled trials, you actually start with your population, and then you would have two groups, the treatment group and then the control group, that are randomized. Okay, so by randomizing them, you're actually going to follow them up over time, and then you compare your results. The randomization would take care of any underlying differences between the study groups that you are not aware of. We call those things confounding, so it takes care of that. One of the things also that is an advantage with randomized control trials is that participants can be blinded. So if the participants themselves are blinded, then they can't really influence the results. That's single blinding, which I believe you also heard about in the first lecture. And then you can also have double blinding, where both the participants and the observers are blinded. They don't know which treatment is being given to any of the groups. You can also have even a level of blinding where even the statistician, participants, everyone is blinded in the study. So that way you're not able to influence the results. Say, for example, if you're sitting in the clinic and you suspect you have two treatments, A and B, and you as a researcher think for some reason that treatment A is better, if the next person who comes to be randomized is somebody you know, there may be tendency for you to want to give that person treatment A because you feel that treatment A is better than treatment B. So to minimize all of that, you don't know who gets what, and that way you're able to follow up and compare. The disadvantage of randomized trials, though, is the fact that they can be expensive. They can take a long time. They can take a lot of money, and you can also have ethical problems rising up in randomized control trials. You can see, take for example, in a case where you already have, say, a vaccine that is in the market, and you know that it's efficacious, and you have another vaccine that you want to introduce. It would be unethical for you to have a control group where you give them a placebo that is an inactive ingredient. So that's kind of unethical because you already know that there's a vaccine that would work. So how would you do that? So sometimes you have to face these ethical issues and think about them in randomized controlled trials. An example of a randomized controls trial, again, another study that was conducted here in the Gambia. The population that they looked at were children 6 to 51 weeks old. They were randomly allocated to an intervention was pneumococcal conjugate vaccine, or the other group had placebo. The primary outcome for this study was first episode of radiological pneumonia, and they had several secondary endpoints, which were things like clinical or severe clinical pneumonia, any invasive pneumococcal disease, and all-cause admissions. So from this study, the comparing the outcome in the group that were vaccinated with the active vaccine, PCV, compared to those who had placebo, the vaccine was found to be efficacious. And this vaccine, alongside with other studies that have been conducted in other sites, has been introduced into the EPI program. Let's now look at our population in general and how we go about sampling. So it's important to really define who your target population is. In the previous study that we just talked about, the target population were children under 5 years of age. So from your target population, you would now have your study population. And once you do that, you would now want to select a sample from your study. So by a sample, we mean a subset of the population that you're interested in. And hopefully you should really do well to make sure that the population are representative. You may wonder why it is that we sample. And if you think about it, resources are definitely not infinite. If you have a huge study population, say all children under 5 in a particular village, there may be so many of them that you can't really select all of them. So you select a sample of the children under 5. It would also take a lot of money and also a lot of time and effort to conduct large studies. And so that is why people would do sampling. And it also would help us to have accurate results, especially just making sure that the sample that we take is representative of the entire population. Once you start, it would be important to first establish your sampling frame, which would be the list of potential respondents. So you may want to have a list of all the households to see which households you may want to be looking at. You may have class rosters, or an electoral register may also be a sampling frame from which you want to now choose your respondents for your study. There are different types of samples. There's probability samples where each member of the population has a non-zero probability of being selected. These include simple random sampling, systematic sampling, stratified sampling, or cluster sampling. You could also have non-probability sampling where members are selected from the population in some random manner. So you can have convenience sampling, purposeful sampling, and snowballing. Specifically looking at random sampling, you would have each member of the population has an equal chance of being selected. And when you have a very large population, then it becomes difficult really to select each member from the pool. And so bias may be introduced, and we will look at some ways in which you can minimize bias. You can usually generate your list of random numbers from a software, something like Excel, and there are different software that you can use to just generate a list of random numbers that will help you to select your participants. With systematic sampling, you are looking at, you're collecting cases in a very systematic way. So, for example, in a village, you have the list of households, you select a first household, and then you start counting. It's the nth number that you're selecting. Say you decide to select every third household. So once you identify the first one on a particular street, you count every 3 houses and you select one household. That would be systematic sampling. It has an advantage over random sampling in that it possibly could be cheaper and easier to conduct. You can also have stratified sampling. It's commonly used probability method that is superior to random sampling because it reduces sampling error. A stratum is a subset of the population that share at least one common characteristic. For example, males and females. You would identify relevant stratums and their actual representation in the population. You then go on to do random sampling to select sufficient numbers of subjects from each stratum. Stratified sampling is often used when one or more of the stratums in the population have a low incidence relative to the other stratums. You could also have clustered sampling, which is a probability sample in which each sampling unit is a cluster. Examples of clusters include villages or schools, and even households can be clusters. It is useful when a good sampling frame is not available or is costly. For example, you may not have the list of everybody in a village, but you may have a list of all the households within the village. So you specifically choose households first, and then you go to those households and randomly select individuals in the households. The cost of obtaining observation increases as the distance separating the clusters increases. So you need to bear that in mind. There is also what is known as convenience sampling, which is used mostly in exploratory research. It's easy, as the name implies, it's convenient. It's a non-probability method. So it's often used when you have, you're kind of doing preliminary research and you want to estimate, you want estimates without incurring much cost, or when maybe time to select a random sample is difficult to achieve. You also have snowballing sampling, which mostly actually used more in qualitative research. It's a special non-probability method, and it's desired when the characteristics is rare. The technique relies on referrals from initial subjects to generate additional subjects. The cost may tend to be lower. So, for example, if you get into a community and you're asking questions about a particular subject matter, once you start with one person, you ask them to lead you to other key informants, people who they know may be aware of the subject matter. So data collection errors can occur either from the interviewer itself, the respondent, or the instrument that you're using, or even the data collection method. So you can have sampling bias. If you use a non-probability sampling method, such as a convenience sample, it's not systematic, so you can have biases there. If you have a low response rate or a high dropout rate, that would also affect your study. We talked about it when we talked about cohort studies. If the dropout rate in the exposed compared to the unexposed is different, then that would affect your comparison. Usually for response rate, you would expect at least 80% of your population to respond to you. And one way you could do that is to maybe make follow-up calls and try to reach out to people to be able to get them to participate in the study. You can also have non-response bias, which really occurs when people do not participate in your study, when the people who do not participate in the study differ from the people who participate in a very significant way. And so you need to minimize that. You can have something like recall bias that we've talked about, where people just don't even remember what it is. I mean, risk factors, for example, because it has happened in such a long time, they don't remember. So you have to try and minimize that. You could also have measurement errors, mostly relating to the tools that you use. If your questionnaire is poorly designed, say for example the questions are not clear, that would definitely affect the studies. So it's always best to test and to validate the tools. You would have had things like pilot testing a questionnaire just to be sure that the questions you're asking are clear to the people and that people are giving you the responses that you would expect. You can also have data entry error, and when that happens, you realize that the person who's putting in the data in the first place is putting the wrong thing. So best way to avoid that is to put in checks. So you can have range checks, you can have logic checks. For a range check, for example, if you're measuring something like temperature, and you have a temperature of, say, something like 50 degrees Celsius, you know that that's not possible. So you set a check so that when anything comes out of range, it is flagged up. So that would always help to ensure that you're not, you minimize errors as much as possible. There are different methods that we can use for collecting data in quantitative studies. Most of the time we use structured questionnaires and questions. And like I mentioned, these need to be validated so that you make sure that you get the response that you're expecting. Traditionally, we have used face-to-face interviews to administer questionnaires, but increasingly also we see that there's lots of online presence with web-based surveys that are available, and also people use telephone interviews, and I've seen that being done here even in the Gambia. So we would now look at mixed methods research. So we've talked a lot about quantitative research and how you can collect data, how you can make sure that you minimize errors. So let's look at mixed methods research. A mixed methods approach combines the characteristics of both quantitative and qualitative research in the same study. So it allows researchers to validate their findings and to verify if the results observed using both methods are complementary, or if they're able to explain any unexpected results from one method or the other. A mixed methods research design is useful in case of research questions that cannot be answered by either a quantitative research or a qualitative research alone. It may require more effort and may require also more resources. There are certain rationale for using mixed methods. For example, it can obtain different multiple perspectives. It can build comprehensive understanding on the subject matter. You can use it to explain statistical results in more depth. You can have better contextualized measures. You can track the progress of a program or an intervention, and study patient-centered outcomes and stakeholder engagement can be explored further. So with mixed methods, there are three basic ways in which this can be done. You can have a convergent parallel design in which you conduct the quantitative data collection and analysis at the same time of conducting the qualitative data collection and analysis, and then you compare or relate the results and try to interpret that. So you discuss areas of convergence or divergence between the qualitative and quantitative results. You could also have explanatory sequential design, where you first do your quantitative data collection and analysis, and you follow that up with a qualitative data collection and analysis. You want to determine what qualitative results need to be further which need further explanation. So once you do your qualitative data, you go on now to follow up with qualitative data just to get further understanding of what you're studying. You can also have exploratory sequential design, in which you then start with a qualitative data collection, and then that builds onto your quantitative data collection. So you're trying to use the qualitative data to develop an instrument that would be used in the quantitative data collection before you go on to interpret your results. Here is an example of a mixed method study that was done here in the Gambia. It was looking at community perspectives on treating asymptomatic infections. I'll encourage you, you can look at the article itself. It was an exploratory sequential mixed method. So the researchers started with a qualitative data collection where they involved in-depth interviews, which we talked about when we talked about qualitative studies, and they also had participants' observations. And so based on what they found, they now went on to do a quantitative data analysis using a cross-sectional study. And these were all done before the trial that was planned. So there was a trial that was planned, which was a clustered randomized trial, where they were looking at reactive treatment for contacts of clinical malaria living in a particular compound. So this was exploratory sequential methods of mixed method. I'll end here with this slide that really talks about reporting statements. If you're interested, you could go further to look at these. So there are different study designs, like we mentioned, and researchers have come up with like standards or guidelines of things that you would expect when you conduct each of these studies. And usually by the time you're publishing, you would have to be sure that you have met the criteria for each of these reporting statements. Say, for example, for observational studies, there's the strobe. So it tells you all the different things that you need to look out for when you're doing observational studies. For randomized control trials, there's the consort. So each of them, starting from your title right out to how you select your participants, how you do randomization and allocation, these all need to be catered for. So you can look out for these reporting statements and help you to understand and get deeper understanding of study research. Thank you.